Hello, I'm Jan Leifman.
And I am Muskan Joshi.
Welcome to Med News Week conference, where we feature presentations by medicine's global leaders. Today we have an amazing keynote speaker in Dr. FIshman. Dr. FIshman is the director for Transplantation, Infectious Diseases, and Compromised Host Program, and is the associate director at the Massachusetts General Transplant Center at Massachusetts General Hospital.
In today's presentation, Dr. FIshman discusses the latest developments and risks associated with xenotransplantation.
Did you know that Dr. FIshman has directed multiple clinical trials for innovative therapies for PCP infection in stem cell and solid organ transplantation, atovaquone, and of new antiviral and antifungal agents, including ganciclovir, valganciclovir, and posaconazole? He's also co-investigator of the first studies of induction of immune tolerance for renal transplantation at the Massachusetts General Hospital, and also developed clinical protocols for transplantation into HIV-infected individuals and the National Multi-Center Clinical Trials in Organ Transplantation to monitor viral infections using novel multiplexed viral molecular assays.
What an inspirational pioneer in the field of transplantation.
So let's tune in and learn from this global leader.
Let's talk about organ transplantation just a little in general, because it's where I spend a good part of my time. There's a growing problem of organ shortages, not just in the United States, but worldwide for treatment of end-stage organ failure. On the average, 20 people die each day waiting for an organ to be available for transplant, and many more worldwide. Over 100,000 people are waiting at present, today, for an organ transplant, but over almost 7,000 died last year while waiting for an organ to be available for transplantation. There are alternative therapies for organ failure, but none of them are as good yet as organ transplantation. There are gene therapies. There certainly are some good antiviral treatments. We're improving organ transplantation and survival. We're starting to think about artificial organs and engineering organs in the laboratory. But the question has always been, where do we get a source of organs for treatment of end-stage organ failure, for people who have heart, lung, liver, kidney, pancreas failure? And where do we get those from? And it's always been considered that animal organs might be possible, but the barriers to transplantation, both immunologic and functional, metabolic, have always been quite high. And so over the years, going back to the 1600s, people have thought about using animal organs to cure humans with end-stage organ disease that would otherwise kill them. And I'm not going to go through the timeline, but excitement has grown tremendously in the last year or so as we have started to see the use of pig organs, both in studies of deceased donors, people who've generously volunteered their bodies for science, as well as a single heart recipient at the University of Maryland who got a heart transplant from a genetically engineered pig not so very long ago. There is a problem with this, and this is the whole balance between why pigs and the risks of using other species. Pigs have always been thought of as useful for this particular purpose because they're well described. They've been developed for commercial use. They can be sized to be used in humans. And even before we learned how to do genetic manipulation, we had experience with inbreeding genetic manipulation in herds. And therefore, you could study the effects of various manipulations in, for example, in primate studies. Over the years, many reagents, laboratory reagents, had been developed to enhance studies of pigs for potential xenotransplantation. With the development of CRISPR, with the ability to modify the genome of pigs, they were able to derive new strains of swine with certain characteristics that were both beneficial immunologically in terms of graft rejection and functionally, that would make them last longer. My own lab and others had developed sequence data for some of the important viruses, and there are also some veterinary labs that have developed experience, and pig organs happen to be resistant to human infections with hepatitis B, hepatitis C, and HIV. So there were some advantages, but there were some disadvantages that have been taken care of largely through the genetic manipulation. So we were poised at a place where we're starting to think that this might in fact be practical. And so how do we approach these kinds of challenges? Well, the way we do it is we think about a package. What we think about is every individual with end-stage organ failure needs a specific organ, a heart or lung or a liver or kidney. They need certain kinds of function, certain sized organs, and they need organs that we think are safe, that have been bred with certain characteristics that don't have bad organisms that might cause infection in the recipient. Coupled with that, all of these animal organs have been studied in preclinical models in non-human primates, in macaques or baboons, to show that they function properly. And therefore, we have already studied the immunosuppressive regimens, the regimens used to prevent graft rejection in those preclinical models. And so this package, if you will, are all the pieces that we put together to think about whether or not we can make xenotransplantation safe. And so what we're trying to do is define parameters. What would make it safe? Well, we have a lot of experience. On my own, I have 35, 40 years of experience taking care of transplant patients. And what we've learned from those is that you need specific assays, you need ways of monitoring infections, you need to be able to follow the patient long-term, you need to be able to adjust their immune suppression. And then over time, you get a synergistic package, if you will, for each of our transplant patients. And over time, we've also come to understand the science of human-to-human transplantation as we're starting to understand the transplant models of xenotransplantation. Now, one of the things I've been teaching or trying to teach for years is that it's not rocket science, that this risk for infection depends on how well your immune system works and how much or what type of organism you're exposed to. So let me give you something that you heard of for sure. Let's talk about COVID. So if you talk about COVID, if you get a big dose of the virus, you're likely or more likely to get sick. If I then alter your immune system so you can't prevent infection, you're even more likely to. So if you're older age, or if you have heart disease, or if you have other kinds of dysfunctions, you're more and more likely to get infected. Well, same is true in transplantation. And so we have individuals where we've caused the immune defect because of the fact they need immune suppression to prevent their body from rejecting or getting rid of the transplanted organ. Why is it such a big concern? Well, think about it. If I have an organ that is carrying infection and I implant it into a body, we're bypassing all of the defense mechanisms that you normally have to prevent you from getting infected. Because it's really a foreign type animal, we may have to increase the intensity of immune suppression. And that organ will supply a culture plate or an ecologic niche for infection inside the body. More than that, many of the organisms that are present in veterinary science haven't been studied in humans. So we don't know exactly how they're going to behave. And further, we've modified the swine genetically, and so that may have other effects on susceptibility to infection. So then the questions are complicated. Can an organism cause infection in a source animal or in a recipient, or persist in a human, or alter genes in the recipient. But it's really not that complicated. It's really a simple idea. Can an organism from pigs set up shop and cause significant infection in immunosuppressed organ recipients? Not rocket science. And so I'm not going to go through the details of all of this, but basically we have sets of organisms that we know from experience can cause infection in immunosuppressed transplant patients. But we have assays for them. We have antiviral or antibiotic agents to treat them. Many of these are infections that we deal with all the time in my clinical practice. There may be some new organisms in pigs that kind of look like organisms that we see in humans. And we say, well, gee, we're a little worried about those. Those shouldn't be in donor pigs. And then the big concern is unknown organisms. So many years ago, we started to look for some of these unknown or novel organisms to raise the question as to whether or not there was a greater risk from xenotransplantation than there was from human to human, or what we call allotransplantation. And I made a list. And this is the list. It's very long. You should memorize it. It'll be on the exam at the end of my talk. No, it really is not such rocket science. There's only one on there, porcine hepatitis E virus, that we know causes infection all the time in people when they get exposed. So we don't want that around. And there are two others, porcine cytomegalovirus and porcine endogenous retrovirus. I'll talk about a little bit more. But in general, it's not as big a concern because most of these organisms can't even infect human cells. So are we concerned? Sure. And that would be reasonable. And therefore, we would put into place a surveillance plan to make sure that a recipient isn't infected. But on the other hand, my concerns have narrowed over the years to a group of organisms that we probably should exclude to make xenotransplantation safe. And so a number of studies have gone on for the last 20 years, and I'll just give you a flavor for some of those. But we need good tests to ask two questions. If I'm screening a pig, has that pig ever been exposed to a virus that might still be present in its body? And those tests are what we call antibody tests or serology. And we use them every day. So if you have a positive serology for chickenpox, it means you've had chickenpox when you were a child. The others are molecular tests that can either tell you you have something in your bloodstream, or because of new technology, we can use what's called metagenomics or high throughput sequencing to look for sequences that we didn't expect to be there. So we have a lot of options in terms of diagnostic tools that we didn't have in the past. What is infection going to look like if we see it when it's transplanted from a pig to a human? Well, if it's a pig-specific organism, it may stay in the xenograft. It may cause disease or may not. It may cause infection of the host, of the human, because it gets out into the bloodstream. The human might even infect the xenograft. Or if you have retroviruses, they may recombine or cause some problems in human cells. So there's a variety of different ways that infection might manifest. And there's a variety of organisms. So for example, we all know that there is a human virus called human cytomegalovirus. Very common. About 80% of the population has been exposed. The pig cytomegalovirus is pretty similar and causes problems in the recipient, including graft rejection and some clotting abnormalities that we prefer not to deal with, but it doesn't infect human cells. There are a number of viruses, including porcine lymphotropic viruses, circoviruses, that would only infect the graft, but not the host. And then there's the porcine endogenous retrovirus. In my lab, cloned porcine endogenous retrovirus type C. But this was discovered during the early eras, back in the 1990s, when AIDS was still an uncontrolled problem, and there was a lot of fear of retroviruses, and we've learned a lot since then. And what we found is that if you combine different types of porcine endogenous retrovirus, or PERV, they can replicate on certain human cells, but only abnormal human cells. And we've never been able to show, despite our concerns, that this virus replicates or can cause copies and infection on normal human cells. The last type of virus that I'll mention is SARS-CoV-2, coronaviruses, or COVID, which human COVID does not infect pig cells, and pig coronaviruses don't appear to infect human cells. So there is a little bit of difference in each one of these viruses. And what we've seen, for example, is porcine CMV infecting pig grafts, and that there are some antiviral agents for porcine cytomegalovirus, but they don't work quite as well. So the question you should ask reasonably is, well, why don't we just get pigs that don't have these viruses? And that's exactly right. That's exactly what we're trying to do, is to breed animals that don't carry potential pathogens for human recipients. So years ago, when we cloned in 1997 the porcine endogenous retrovirus, we found out that they were RNA viruses. They were endogenous viruses, which meant they were in the genome of every single pig. And that two types, PERV A and B, could infect human cells, but not very effectively. PERV C didn't even infect human cells, but if you recombined a little bit of PERV A with a piece of PERV C, they seem to be infectious for human cells of a certain type. And at that time, we said, gee, it's 1998, we're thinking about pig heart trials, but they never happened because regulatory authorities, and I think probably justifiably, said this was a bad idea until we had more information about these endogenous retroviruses that I had helped characterize. So I think this was probably a good thing at that time. So as I've said, these PERV, these porcine endogenous retroviruses, are present in normal swine, but have never been shown to infect human cells. And so they take a little piece of PERV A and a piece of PERV C, and together they're infectious for human cells, and they replicate, they cause infection more effectively. And they can even go back into the genome and reinfect the pig. And that over time, if you put cells in culture, that there are more and more of these PERVs. And so what we were thinking was it would be good to exclude these because we just don't know what they do. Will they enter human cells? Will they cause altered regulation of genes like cancer genes? Will they alter something else in your metabolism? And therefore, it's better not to have it. So what we thought was, could we exclude PERVs from donor swine that would be used in xenotransplantation? One of the things is that normally our cells prevent infection due to retroviruses. And that's because we have a whole group of endogenous factors that prevent infection of human cells. So we have a variety of ways of thinking about reducing the risk of PERV infection. We can choose an animal as a donor that doesn't have these viruses, PERV A, B, or C. We can develop vaccines, although they didn't seem to work particularly well in prevention. We could genetically manipulate the pig's genome to try to get rid of the porcine endogenous retrovirus. We could rely on the natural immunity of human cells because we, in fact, have never seen human infections due to PERV. And if all else fails, all of the drugs we have for HIV, or many of them, appear to have activity against PERV. And so we had a number of options. And then we had a revolution in molecular biology called CRISPR, or CRISPR-Cas9, largely invented by George Church and others. And in collaboration with George and Luhan Yang, we showed that in a cell that made PERV, we could use this CRISPR system to completely get rid of PERV. So that it wasn't a question of 1 or 2 copies. There were 62 copies of this virus in the pig genome, and we were able to eliminate all of them. And so this was the first demonstration that you could cure a cell line of a virus or an endogenous retrovirus. And then Luhan and her team went on to show that you could actually do this in pigs, and therefore we have had pigs develop that are lacking all PERVs, A, B, C, and AC. And so the safety margin gets increased as a result of using this particular strategy. Now, I'd like to take a step back for a second. In human-to-human or allotransplantation, over the years that I've been practicing, one of the biggest advances we've had, for example, is the use of organ donors who have hepatitis C infection. Why is that? Well, we have, as I told you at the beginning, we have a severe shortage of organs for transplantation. And then in the last decade, we've actually developed, and I say we, the pharmaceutical companies and others, have developed therapies for hepatitis C virus. That means we could give somebody an infected organ and then cure them of hepatitis C. We also could use organs where we weren't quite sure whether or not they'd carry infection. And the biggest risks, of course, were for hepatitis C and hepatitis B and for AIDS, HIV. And yet, with the availability of drugs that control those infections, we are able to give individuals an opportunity to survive, to get organ transplants from donors whom we might not have considered in the past before we had these alternative therapies. I call that acceptable risk. And the answer is, if you need a heart or a lung or a liver, where there really aren't alternative therapies, you would accept certain risks with the likelihood that you could be cured of any infection that you might have got. And the rate of transmission of infection in human-to-human transplantation is less than 0.1%. It's quite low. And therefore, using that as a concept, what we say is, if there is a clinical need and we can make it safe, then xenotransplantation should also be acceptable. Okay. So you say, I'm willing to take a pig organ to save my life, maybe for a temporary period of time, or maybe for a longer period of time. But let's assume it's for life. How do we know if there are organisms present there? Well, we screen the source animals. We start looking for lists of organisms before we ever use the pigs as donors for organs. What if we miss something? Well, then we have to monitor the xenograft recipients. So we do blood tests every month, every 3 months, every year to look for potential infections. What if they come in looking like they have an infection? Well, as you all know, and maybe some of you have had COVID, everybody gets infections. Sometimes they're severe. Well, immunosuppressed individuals, transplant patients, both organ transplant, stem cell transplants, are susceptible to a broad range of infections. We see them all the time coming in with infections. And so how do you know if somebody has a pig-derived infection or a human-derived infection or something that they just got from their child who's going to daycare? I don't know how many of you have children in daycare, but certainly that is the biggest source of infection that I've ever seen. So we need ways of screening them. And so we have as a series of assays, of tests, lab tests. And we screen the source animals, we screen the recipients, we test the recipients when they get sick, both for human infections and pig infections. And we might even screen the surgical team and contacts of the animal and of the recipient, just to be on the safe side. And certainly we'll do that early on in clinical studies. So let's think about this. We're starting to think about clinical trials, and the first pig heart transplant has already occurred. What do you have to do to run a clinical trial? You can't go out and just get a pig organ and start a study. We have to go to the Food and Drug Administration. We have to go through our institutional review committees. We have to go through ethics panels and others. So it's a rigorous process. What are they going to ask us? What they're going to say is, you're going to do a trial of which organ? You say, oh, I want to study livers in people who need liver transplants. And I'm going to screen my pigs for two things. I'm going to screen them for organisms that may cause infection after transplant. And I'm going to make sure that those organs will work in the human environment so that things like clotting factors, things like uric acid, and that they won't get gout and they won't get heart disease, and the cholesterol will be normal, and a variety of other important metabolic functions. And then I'm already will have studied the immune suppression, the drugs I need to give to prevent graft rejection, because I've already studied these organs in non-human primate models. And that package, if you will, which is what I started with, is what we think about when we're thinking about a clinical trial. But the key is to keep our eye carefully on the recipient, on the patient, and say, what can we do to make this as safe as is possible? How can we be the most rigorous we can be in developing these trials? And I've already suggested to you that the risk of infection is not zero. It's not zero for any immunocompromised host. Therefore, we've started to develop diagnostic tools and therapies for potential infections. We should expect that people who are immunosuppressed, when they go out into the community, they're going to get flu and they're going to get other infections that are just circulating in the community. There may be some unexpected swine pathogens, and therefore we're going to use modern genetic techniques I've already mentioned, metagenomics or high throughput sequencing, to look for unknown pathogens. We're going to save baseline samples of cells and sera so that we can go back and look at them to see if new infections have occurred in the future, and that they'll be available for public health authorities. There are some advantages, as I've mentioned, to porcine xenografts in the fact that they will be resistant to some common human organisms. But in fact, we don't know exactly what's going to happen until we do clinical trials. And there's a little bit of faith that has to be there, which is that we're going to do the best we can, but we can't know everything. And so we're going to be very rigorous and we're going to be very careful as we go into clinical trials. So we want to be careful, but we don't want to be foolish in terms of the potential for infections in the immunocompromised xenograft recipient.